Very well, one can move the world, one can melt iron by the power of words. So I would like to invite Deepika for delivering a speech on the topic of nucleus. She is going to give so much information about nucleus, so listen her carefully.
Respected class in charge and my dear friends, a very great morning to all of you.
Very great morning, Deepika.
Myself Deepika. I am the student of class 11th Science standing before you for delivering a speech. And my speech topic is nucleus. Nucleus, the word with which we students are very familiar with. We all know that it is the brain of a cell. It is the control center of a cell which controls all the metabolic activities and functions of a cell. So today in this session we are going to know more about nucleus. Nucleus was first discovered by Robert Brown in 1831. At that time its word was given from the word nucleolus, which means seed. After that, many scientists were very curious to know that what is nucleus, what does it do. They were very curious to know about its structure. But at that time, only simple microscope was known. So it was not easy for them to know the very small structure of nucleus because its size is about 10 raised to power minus 15 meter. And that's very small to know under a simple microscope. When electron microscope was discovered, then scientists found that nucleus is a double membranous structure and its membrane is known as nuclear membrane as well as nuclear envelope. It is, as I have told you, a double membranous structure, which means have two outer membranes. That first one is the outer membrane and second one is the inner membrane. They are separated by a distance and have some space between them. That space is known as perinuclear space. And these membranes forms a barrier between the cytoplasmic material and the material present inside the nucleus. They are the barrier. Now about interphase nucleus. What do we mean by interphase nucleus? It is the phase of nucleus when it is not dividing. Which means when the nucleus is not dividing, at that stage we will call it interphase nucleus. And at that time it have three main things in it. The first one are chromatins. Second one is the nuclear matrix, also known as nucleoplasm. And the third one are nucleoli. Plural we will call it nucleoli and singularly nucleolus. One by one, I will tell you about all these three things. First one, I will tell you about nucleoplasm. Nuclear matrix, it is the matrix or the fluid present inside the nucleus, which contains chromatins and nucleoli inside it. Second one is the nucleoli. It is the dense part of the nucleus, which is the site for ribosomal synthesis as well. And if this nucleoli is present in larger amount and more nucleoli are there, then it also helps to carry out protein synthesis, which means it is the site for synthesis of two things, that are RNA, ribonucleic acid, as well as proteins. The third one that I have told you was chromatin, are the extended and elaborated nucleoplasmic fibers. They are the nucleoplasmic fibers that contain DNA, RNA, some basic proteins named as histone and some non-histonic proteins as well. These four things are present in chromatin. Now, the cell at many stages show some long thread-like structures that are named as chromosomes. These chromosomes contains all the hereditary information of the cell of the parent body. They are transferred from the parent to the progeny and they are present in 23 pairs. Means if we will count them singularly, they are 46, but as they are present in pairs, so they are 23 pairs. In a human body, when a child is born, he gets 23 chromosomes from the mother cell and 23 chromosomes from the father cell that combinedly form 23 pairs of chromosomes. Now, chromosomes have a centromere in their structure that helps in cell division as well, and centromere is surrounded by kinetochores that are disc-shaped structures. Disc-shaped structures around centromere are known as kinetochores. They also help in cell division. And on the basis of position of centromere, chromosomes are of 4 types. The first one is metacentric. Second one is submetacentric. Third one is acrocentric, and fourth one is the telocentric chromosomes. The first one, as the name suggests, metacentric, which means the centromere is present in the center. Both the arms of the chromosomes are equal. The second one, submetacentric. In this, the centromere is not located just at the center. It is present just above the center. Means one arm is longer than another arm. So there is a difference between their lengths. Third one, the acrocentric. Acrocentric chromosomes are those chromosomes in which the centromere is present close to one end. Not at the end, just close to one end. In these chromosomes, one arm is very longer than the another arm. The fourth one, but not the least, telocentric. Telocentric chromosomes are those chromosomes in which the centromere is present at the terminal, means having one longer arm. They have some primary constrictions as well and secondary constrictions known as satellite as well. And the repeated sequence of DNA on the satellite is known as satellite DNA. So I hope you get to know what I meant to tell you, and that's all from my side. Thank you and have a nice day.